Hi, I'm Prithviraj Bose. I'm a professor in the Department of Leukemia at the MD Anderson Cancer Center in Houston, Texas. And today I'm going to be talking about novel therapies for high-risk MPNs, in particular myelofibrosis. This will be a very quick overview. There's a lot going on in the field, but I'll try to at least do some justice to all the exciting agents and sort of give you a little flavor of what is coming. So at ASH 2023, we of course had two phase 3 trials read out, and these were both ruxolitinib-based combinations. There was pelabresib, the BET inhibitor, and there was navitoclax, the BCL2 BCL-XL inhibitor. These were compared to ruxolitinib and placebo in frontline, which means JAK inhibitor naive patients with myelofibrosis. Now, the trials had somewhat mixed results. The pelabresib trial was the larger trial, and actually both trials, I should say, showed a near doubling or exact doubling in the case of navitoclax, but a near doubling of the spleen response rate at 24 weeks. So that clearly was met, and that was the primary endpoint of both trials. However, the story was a little bit different with symptoms. With the navitoclax combination, the symptoms were not only not better than ruxolitinib and placebo, they were actually numerically a bit worse. And there was clearly no symptom benefit in that trial. Now, in the pelabresib trial, which was, again, remember, the larger trial, there was not a statistically significant symptom benefit, although the p-value was agonizingly close at 0.0545. And this was about the absolute change in the TSS. If you look at the mean TSS and average it out, the absolute change in the mean TSS between the two groups favored the pelabresib arm over placebo, but not quite statistically significantly. Numerically, yes. And then also the TSS50, which is the more familiar traditional endpoint that has been around for quite a while, that too numerically favored the pelabresib arm, but not statistically significant. So those really were the main messages from those trials. Again, the pelabresib was the larger trial, and also there were some other differences. For example, the pelabresib trial had way more intermediate 1 patients, DIPS intermediate 1, whereas the navitoclax trial had primarily intermediate 2 and high risk, and they used DIPS plus rather than DIPS. So kind of small nuances, but I think can be important as we try and evaluate these results. And then also in the pelabresib trial, you started at a lower dose of ruxolitinib than the platelet count would indicate. And then you went up. You had to go up if certain parameters were met, but you did not start at the labeled dose of ruxolitinib. This again was different from navitoclax, where they started at the labeled dose of ruxolitinib. One last thing on these trials, the pelabresib trial, which again, frankly, in my opinion, is the more impactful of the two trials, did show more than a doubling of the proportion of patients who had both a spleen response and a symptom response at 24 weeks. So that percentage went from 18.5 to 40.2 for people meeting both TSS50 and SVR35 at 24 weeks. Well, and then there is Selinexor. Selinexor is, much earlier in its development, but it is in phase 3 now as well, based on a very highly promising phase 1 study in which in 14 patients, the dose of Selinexor of 60 milligram once a week, which is a lot lower than, for example, the approved dose in myeloma, was studied in combination with ruxolitinib. And in these 14 patients, again, small numbers, but there was a very striking spleen response rate of 92% in the evaluable population, 78 in the ITT population, and the symptom response rate was 78 in the evaluable population and 58 in the ITT population. So clearly, regardless of how you look at it, very promising numbers, and so this combination is going into a placebo-controlled phase 3 trial in frontline patients, JAK inhibitor naive, with myelofibrosis. Platelets, of course, have to be 100 or higher for all of these frontline combos. And then really quickly, there are, of course, add-on strategies that continue to be evaluated. The navtemadlin compound is MDM2 inhibitor from Cartos. There were exciting results from this at EHA of last year. Nothing new on this at ASH, but in the add-on setting, it gave you a spleen response rate and a symptom response rate of 32%. Which is actually pretty good, if not the best, in this add-on setting where you have a suboptimal responder to Rux alone and you add another drug, and 32 and 32 is about as good as you get. And then there is also bomedemstat, the LSD1 inhibitor, better known for its results in ET. But at ASH 2023, there was an oral abstract on this, looking at the combo of bomedemstat and ruxolitinib in both frontlines, so JAK inhibitor naive, and in the add-on setting. So it was looked at in both these settings, and generally what emerged was that for actually both spleen and symptoms, in the patients that were frontline, the response rate was about 33%, and in those who got the bomedemstat as an add-on, the response rate was about 20%. And then there are single agents that continue to be evaluated, not in combo with the JAK inhibitor, at least not now. There is imetelstat, which is the telomerase inhibitor. This is in phase 3. We haven't heard anything new on this in just a little while. Of course, there is a lot of excitement around this drug and its potential approval for lower risk lower risk MDS, perhaps later this year. But then in myelofibrosis, there is an ongoing phase 3 in the JAK inhibitor failure setting, where imetelstat is compared to best available therapy, which does, however, exclude JAK inhibitors. And the primary endpoint of this trial, based on some signals we saw earlier with this drug, is overall survival. There is a PIM kinase inhibitor from Sumitomo, which is interesting. Some nice results have been shown on this, being evaluated currently, at least, as a single agent in the post-JAK inhibitor or JAK inhibitor ineligible or intolerant setting. And in this group of patients, in particular, it has had a symptom benefit more pronounced than a spleen benefit, although that's there too. And then furthermore, the symptom benefit is actually correlated with cytokine decreases. So something to watch that certainly looks interesting. I'll wrap up finally with a lot of therapies on which we don't necessarily have clinical data, but we do definitely have some very interesting mechanisms of action. And some of these are actually cellular and immunotherapies. So one is the regulatory T cells. These are being added in patients with ruxolitinib, patients who are on ruxolitinib at a stable dose, but just have a suboptimal response. And the regulatory T cells derived from umbilical cord blood are being added. Some preliminary efficacy has been presented, very safe. So this is something that is ongoing. And then very much in vogue currently is this idea of targeting the mutant driver as opposed to the wild type. So there are strategies, for example, against mutant calreticulin, such as an antibody strategy from Incyte and a T cell redirecting antibody from Janssen, which is really a bispecific that uses CD3 positive T cells to mount an immunologic attack against the mutant calreticulin. Now remember that mutant calreticulin is particularly amenable to immunologic targeting because it actually comes outside the cell and associates with MPL or the thrombopoietin receptor on the cell surface. And this really is both integral to its oncogenic signaling and also is a therapeutic target. It's a therapeutic vulnerability because now it's exposed to the immune system and one can try and target it that way. And then there are mutant selective JAK2 inhibitors coming. Insight has one of those. The preclinical data were presented at the last ASH meeting. So again, a mutant selective JAK2 inhibitor. So just hitting JAK2 V617F by binding to the pseudokinase domain, which is certainly something very different from all the JAK inhibitors we currently have. And the obvious theoretical advantage of a mutant selective JAK inhibitor would be that it would not cause the cytopenias that we associate with targeting JAK2 as a whole, which means targeting wild type as well as we do now with drugs like ruxolitinib or fedratinib. And then finally, there is something called a type 2 JAK2 inhibitor. And this comes from the work of Ross Levine from many years ago, where he and his laboratory colleagues showed that the current class of JAK inhibitors, which are called type 1 JAK inhibitors, can remain bound to the target, which is to the enzyme, the kinase domain of JAK2, but the signaling through the JAK-STAT pathway can nevertheless proceed, can continue. By something called heterodimerization, where, say, JAK1 could team up with type 2 or JAK3 and still signal despite the blockade of JAK2. To overcome this, there is now, from a company called Ajax, a drug that is a type 2 JAK inhibitor, which actually binds to and stabilizes the inactive conformation of JAK2, not the active conformation that all the current JAK inhibitors bind to. So again, all of these therapies, the mutant CALR antibody, the T-cell redirecting antibody against mutant CALR, the mutant selective JAK2 inhibitor, the type 2 JAK2 inhibitor, are all just entering the clinic with no data available, no data that has been presented. Really too early for all of these, but certainly very, very promising. And again, there is a whole another section that I couldn't cover in this short overview, which is the drugs for anemia. And there are several of those as well.